Dear students, after studying this module, you shall be able to know about applications of Y-chromosome testing, structure of Y-chromosome, different profiling systems for Y-chromosome testing, and gender typing. We shall start with the introduction to Y-chromosome. The Y-chromosome is inherited from the father and is conceded onto all male offspring. Thus, the Y-chromosome is distinctive to males. The chromosome encodes dozens of genes essential for male-specific functions, including sex determination and spermatogenesis. Y-chromosome loci are very important for forensic DNA profiling. And in this module, we shall be discussing about the various such applications related to Y-chromosome testing. For instance, the Y-chromosome STR, that is YSTR, used in forensic DNA testing is male-specific and is thus useful in investigation related to sexual assault where male suspects are involved. The evidence that is collected in such cases usually consists of mixtures with high levels of female DNA and low levels of male DNA. The Y-chromosome specific loci can thus be examined without interference from the large amounts of female DNA. Furthermore, the YSTR system is useful for determining the numbers of male perpetrators involved in sexual assault cases involving more than one male. The YSTR loci used for forensic applications are located in the non-recombining section of the Y chromosome so that the paternal lineages can be established. The technique can be used for paternity testing and identification of missing persons. Finally, data interpretation can be simplified by the use of a single allele per YSTR locus profile. Figure, pedigree showing patrilineal inheritance where all shaded males have the same Y chromosome barring any mutations. Now we shall discuss about the applications of chromosome Y testing. Y chromosome DNA testing is vital for a number of diverse applications of human genetics, including forensic evidence examination, paternity testing, historical investigations, studying human migration patterns throughout history, and genealogical research. In terms of forensic applications, there are both advantages and limitations to Y chromosome testing. First, we shall discuss about the advantages of Y chromosome testing. First, male-specific application can enable examination of a male perpetrator's profile even in mixtures with high levels of female DNA in sexual assault cases. Second, additional mixtures may possibly be analyzed. Example, fingernail scrapings, saliva on skin, etc. Third, paternal transmission from a father to all of his sons extends possible reference sample providers and enables tracing family lineages. Now we shall discuss about the limitations of Y chromosome testing. Since paternal relatives are indistinguishable, YSTR typing cannot be used to differentiate among siblings or even distant paternal relatives. Second, without recombination between loci, the product rule cannot be used and thus the discrimination power of YSTRs is limited by the size of the population database used. Third, duplications and deletions can cause difficulties in the analysis. The primary value of the Y chromosome in forensic DNA testing is that it is found only in the males. The SRY, that is sex determining region of the Y chromosome, gene determines maleness. With chromosome Y tests, interpretable results can be obtained in some cases where autosomal tests are limited by the evidence, such as high levels of female DNA in the presence of minor amounts of male DNA. These situations include sexual assault evidences from azoospermic or vasectomized males and blood-blood or blood-saliva mixtures where the absence of sperm prevents a successful differential extraction for isolation of male DNA. In addition, the number of individuals involved in a gang rape may be easier to decipher with Y chromosome results than with highly complicated autosomal STR mixtures. Using chromosome Y specific PCR primers can improve the chances of detecting low levels of the perpetrator's DNA in a high background of a female victim's DNA. Y chromosome tests have also been used to verify amelogenin Y deficient males. The similar feature of the Y chromosome that gives it a benefit in forensic testing, namely maleness, is also its principal limitation. A bulk of the Y chromosome is transferred directly from father to son without recombination to shuffle its genes and provide greater genetic variety to future generations. Random mutations are the only mechanism for variation over time between paternally related males. Thus, while exclusions in Y chromosome DNA testing results can aid forensic investigations, a match between a suspect and evidence only means that the individual in question could have contributed the forensic stain, as could a brother, father, son, uncle, paternal cousin, or even a distant cousin from his paternal lineage. Needless to say, inclusions with Y chromosome testing are not as meaningful as autosomal STR matches from a random match probability point of view. On the other hand, the presence of relatives having the same chromosome Y expands the number of possible reference samples in missing persons investigations and mass disaster victim identification efforts. Chromosome Y testing also aids familial searching. Deficient paternity tests where the father is dead or unavailable for testing are benefited if chromosome Y markers are used. However, an autosomal DNA test is always preferred when possible since it provides a higher power of distinction. The Y chromosome has also become a popular tool for tracing historical human migration patterns through male lineages. Anthropological, historical, and genealogical questions can be answered through chromosome Y results. Now we'll see some of the uses and advantages of Y chromosome testing. First, uses forensic casework on sexual assault evidence. Advantage: male-specific amplifications can avoid differential extraction to separate sperm and epithelial cells. Second, verification of amelogenin Y deficient males. Advantage: analysis of multiple regions along Y chromosome that should not be affected by deletion of the amelogenin region. Most important, paternity testing. The advantages: male children can be tied to fathers in motherless paternity cases or testing of male relatives if father is unavailable. Next is missing persons investigations. Advantage: patrilineal male relatives may be used for reference samples. Next important use is human migration and evolutionary studies can be conducted. Advantage: lack of recombination enables comparison of male individuals separated by large periods of time. And one more important use is historical and genealogical research. The advantage is surnames usually retained by males can make links where paper trail is limited. Now we'll study about the structure of Y chromosome. A detailed analysis of the finished reference Y chromosome sequence was described in the 19th June 2003 issue of Nature by researchers from the Whitehead Institute and Washington University. At 50 MB, the Y chromosome is the third smallest human chromosome, only slightly larger than chromosome number 21, which is 47 MB, and chromosome number 22, which is 49 MB. In this figure, we can see the structure of human Y chromosome. The non-recombining region of the human Y chromosome, that is NRY, comprises approximately 95% of the chromosome. The two tips of the Y chromosome, known as pseudo-autosomal regions, that is PAR, recombine with X chromosome homologous regions. PAR1, located on the tip of the short arm, that is YP, of the Y chromosome is approximately 2.5 MB in length, while PAR2 at the tip of the long arm, that is YQ, is less than 1 MB in size. Skatlsky et al. In 2003 renamed the NYR the male-specific region because of evidence of frequent gene conversion or intrachromosomal recombination. A total of 156 known transcription units, including 78 protein coding genes, are present on MSY, that is male-specific region. The Y chromosome is highly duplicated either with itself or with the X chromosome. Three classes of sequences have been categorized in the Y chromosome: X transposed, X degenerate, and ampliconic. Now we'll study about the human Y chromosome genome. The human Y chromosome genome contains approximately 60 million base pairs, and the chromosome can be divided into two regions: the pseudo-autosomal region, that is PAR, and the male-specific Y region, that is MSY. First, we'll see a pseudo-autosomal region, that is PAR. Approximately 5% of the Y chromosome sequence is located at the telomeres of the chromosome. In particular, PAR is located at the tip of the short arm and PAR2 is located at the tip of the long arm. This region undergoes recombination with homologous region of the X chromosome during meiosis in males. In this figure, we can see the schematic diagram of X and Y sex chromosomes. And in the B portion, we can see the Y chromosome is composed of both euchromatic and heterochromatic region. Second is male-specific Y region. The remainder of the Y chromosome is known as male-specific Y region. It was previously called the non-recombining Y region or the NRY region. It does not participate in homologous recombination. However, certain sections involve intrachromosomal gene conversion. About 40 megabases within the MSY region are heterochromatic, highly repetitive sequences, including the centromeric region and the bulk of the distal long arm. The euchromatic region is about 23 MB, and most of it has been sequenced. Certain sections of the euchromatic region share some homology with the X chromosome. For instance, X transposed sequences of the Y chromosome are 99% identical to sequences within XQ21, a band in the long arm of the X chromosome. Additionally, dozens of genes located in the euchromatic region share 60% to 96% homology with their X chromosome counterparts. These X homologous regions should be avoided when selecting chromosome-specific markers for DNA profiling. Third is polymorphic sequences. The Y chromosome contains an abundance of repetitive elements, namely STRs, ALU and line elements. Many of these are highly polymorphic. To date, YSTRs are usually used for Y chromosome DNA testing. Single nucleotide polymorphisms, that is SNPs, at the Y chromosome are also useful for forensic applications. Now we shall discuss about the various profiling systems for Y chromosome. First is YSTR. More than 400 STR loci have been identified in the Y chromosome genome. The precise locations of these loci have been sequentially mapped using human genome sequencing data. The distribution of YSTR loci at the Y chromosome has also been analyzed. Most YSTR loci, approximately 60% of the 400 identified, are located on the long arm of the chromosome. About 22% are located on the short arm, and a few are found in the centromeric region. YSTRs in the telomeric region have yet to be identified. YSTRs have been analyzed. Among 400 YSTRs, 60% are dimeric repeats, 39% are trimeric, 45% are tetrameric, 9% are pentameric, and 1% are hexameric. Fewer than half the STRs have been characterized. Some loci are polymorphic and are useful for forensic applications and developing new YSTR multiplex systems. The STR loci of the Y chromosome are usually referred to as haplotypes. A haplotype is a collection of alleles that are usually linked, inherited together, since homologous recombination does not occur on the majority of the Y chromosome. The most commonly used YSTR loci for forensic testing are DYS19, DYS385A and B, DYS3891, DYS3892, DYS390, DYS391, DYS392 and DYS393. The following figure shows human cytogenic map of Y chromosome. The YSTRs and positions are also shown. Next profiling system is core YSTR loci. In 1997, the European minimal haplotype, that is EMH locus set, was recommended by the International YSTR User Group for forensic application. This haplotype set includes a core set of YSTR loci. These are DYS19, DYS385A and B, DYS3891, DYS3892, DYS390, DYS391, DYS392 and DYS393. In 2003, the US haplotype loci were recommended by the Scientific Working Group on DNA Analysis Methods, that is SWGDAN, for forensic DNA analysis. The US haplotype loci included the EMH loci set plus 2 additional loci, that is DYS438 and DYS439. DYS385 and DYS389 are multilocal YSTR loci, MLL. The MLL designation refers to a presence of a particular STR at more than one site on the Y chromosome DNA due to duplication. To date, about 50 such MLL YSTRs have been identified. Further MLL subdivisions are designated are bilocal, trilocal, etc. DYS385 and DYS389 are bilocal. The DYS385 locus has 2 inverted duplicated clusters and is separated by a 4 into 10 raised to power 4 base pair interstitial region. It can be amplified by a single set of primers. One allele is observed if the duplicates are the same length. If the duplicated clusters have different lengths, they can generate 2 different alleles when amplified. The smaller size allele is designated A and the larger size allele is designated as B. The DYS389 locus has 2 duplicated clusters with the same orientation. In a single set of PCR primers, there are 2 binding sites for the same forward primer at each 5' flanking sequence of the core repeat region DYS389. These binding sites between DYS3891 and DYS3892 are about 120 base pair apart. Therefore, 2 amplicons are produced. DYS3891 is designed for the smaller allele and DYS3892 is designated for the larger allele. The average mutation rate for the core YSTR loci is approximately 10 raised to power minus 3 per generation, similar to the mutation rate of autosomal STR loci. Mutations can exert major impacts on the interpretation of paternity test results. All this has been described in this figure. Next profiling system is multiplex YSTR. The application YSTR for forensic casework was initiated in Europe. In the US, the laboratory of the Office of Chief Medical Examiner in New York City was the first to perform YSTR testing of 4 loci, that is DYS19, DYS385A 390, DYS 389-1 and DYS 389-2. The use of YSTR loci has been facilitated by various commercially available PCR amplification kits in multiplex systems. Relagene Technologies developed the first commercial multiplex YSTR system, the Yplex TM6. The kit includes DYS 19, DYS 385A and B, DYS 389-2, DYS 390, DYS 391 and DYS 393. Additional commercially available kits with more YSTR loci are now available and have been validated for forensic use. To improve discriminating power, multiplex systems including new YSTR loci are desired. Many new YSTR loci are being characterized for developing new multiplex systems. In the figure, we can see the result from the commercial YSTR kit by Yplex TM6. Now we will discuss about gender typing. Gender typing of a biological sample is useful in forensic investigation. For example, for victim identification in disaster cases and suspect identification in sexual assaults. One commonly used gender typing marker is the amelogenin or the ML locus, amelogenin locus. This region encodes extracellular matrix proteins involved in tooth enamel formation. Mutations to the ML gene can lead to an animal defect known as amelogenesis, imperfect. The ML locus has two homologous genes. MLX is located on the human X chromosome and MLY is located on the human Y chromosome. Although the genes constitute a homologous pair, they differ in size and sequence. Gender typing can be performed using various primers designed specifically for the sequences of the homologous region of these genes, followed by amplification. Different sizes of amplicons are obtained. The most commonly used gender typing method at the ML locus is the detection of a 6 base pair deletion at intron 1 of MLX. This deletion is not present in MLY. Primer sets were developed to amplify both alleles in a single PCR by Forensic Science Service in the United Kingdom in 1993. The amplicons generated from MLY and MLX are separated by electrophoresis. The observation of the MLX fragment also indicates a female, whereas the observation of both MLX and MLY indicates a male. Nevertheless, primate and some rudiment DNA can be amplified as well, but the amplicon sizes vary. The ML locus has been co-amplified with other markers to provide a combined gender and identity test. Such combined tests have been used in D1S80 AFLP and various STR multiplex analysis. Next is MLY null mutations. Several cases of MLY null mutations have been reported. Only the MLX fragment was detected in these MLY null males. Many of them are phenotypically normal but present the ML gender types of females. Various interstitial deletions at the Y chromosome short arm have been identified as the cause of some ML null gender typing. The frequency of MLY null males is rare but is higher in Sri Lanka and India. Now we'll conclude this module with the summary. In this module, we learnt about the Y chromosome and its structure, applications of Y chromosome testing like forensic casework on sexual assault evidence, verification of amelogenin Y deficient males, paternity testing, missing persons investigations, human migration and evolutionary studies, human Y chromosome genome like the pseudo-autosomal region, male-specific Y region and polymorphic sequences, different profiling systems used for Y chromosome such as YSTR, core YSTR loci, multiplex YSTR. Gender typing is also used in forensic investigations such as identification in mass disasters and suspect identification in cases of gang rapes.